Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Okay, thanks, Dean, for the introduction. And this is a talk about BioMashups. It's joint work with the people listed there. I'd just like to acknowledge Jiro Sumitomo, who did most of the initial exploration, and Felicity Newell, who did the work on the protein case study, which we'll talk about towards the end of the talk. So here's a broad agenda for the talk. We're going to talk about bioinformatics, and then a little bit about a kind of an ad hoc taxonomy of workflows, where we're talking about a distinction between exploratory and routine workflow, and fairly obviously I think mashups will come into the exploratory category. At just what level of granularity is of course one of the purposes of the talk. So we'll then give a brief introduction to mashups and biomashups and find some unexpected images of Tony Hay, so we'll look at that a little bit later on. Some basics and some canonical examples. We'll look at some limitations, and then of course we'll get somewhere towards the future. So bioinformatics, for this audience, I don't really need to emphasize the fact that there's a rather major diversity of tools and data sources, traditionally web-based interfaces and some standalone applications, more recently web services hooks, and of course these things need to be combined in some way to do anything serious. And of course part of this entire agenda is about making it easier for the scientists to do that. So what we're interested in at this stage then is linking tools together, and linking tools together in a way which enables scientific discovery. And so the trade-off that we're traditionally faced with in this domain is one of workflow sophistication versus development effort. And so it's a choice in many respects between keeping it simple and keeping the scientist involved, and making it more complicated and then requiring the postdoc or the research assistant to do it, and consequently making the head scientist, if you like, the leading scientist, a client of the lab staff. So bench scientists, as we've talked about before, are not software engineers, but as I think Luke pointed out in his talk, if they're given the appropriate tools, they can do an awful lot of good stuff. And mashups is one of the possibilities here. They can chain operations together. They know what operations they want to use, and it's a matter of making that process somewhat more readily available with the appropriate primitives and the appropriate glue. So let's look at two extremes of scientific workflow, and of course the top part of this slide will offend anyone who's had anything to do with scientific workflow because it's being used in the loosest possible sense. So the manual data management system is a cutesy name for cut and paste via Excel. Excel is the data transfer infrastructure. Now, of course, this cannot scale, and it can't be used for anything incredibly sophisticated, but the major advantage that it has is that it has no barriers to entry whatsoever. And so that's, of course, one extreme. At the other end, we have the robust workflow systems like Taverna, Kepler, and more recently Trident. And so these things are, of course, essential for high-end instrumentation. They're very well engineered. There's support for provenance, and they're robust above all else. But the major point here is that there's a significant setup cost and a familiarization burden, particularly for the end-user scientists. Now, none of what we can do in the mashup frameworks that we'll talk about later cannot be done in one of these environments if they're already set up and they're familiar. But what we're arguing, I think, is that the mashups provide an easier pathway for people to get involved in this space. So I've talked about the two extremes here, and so there's a kind of a sweet spot or middle ground here, which traditionally corresponds to what sophisticated bioinformaticians would do via Perl scripting. So the idea here is, Perl or Python or whatever you happen to be using, and there are, however, significant programming skills needed. It's something where people can work out what they want to do manually and then automate it as a kind of a Perl-based or a script-based workflow. But there are, even here, some significant barriers for using it for ad hoc experimentation. There are, of course, quite quite significant programming skills required, and those programming skills aren't necessarily possessed by all bench scientists. There's also just the burden of actually gluing the things together using scripts, even for those of us who are computationally sophisticated. It can be a pain to do this. And so what we're questioning here is really whether this is a niche for what we come to call biomashups. So what are mashups? Well, mashups is a very broad term. It means a lot of things. It means a lot of different things to a lot of different people. But fundamentally, they're web-based applications for the combination of data sources and services. Initially, they were ad hoc scripts, essentially, via JavaScript to unify a bunch of services and tools, services and data sources. Flickr, Facebook, things like this, mashups on top of those are probably the most commonly known. But they have, in principle, the same issues of Perl scripting. But in their initial form, they had this additional burden of the need to organize hosting for the web mashup. And so in the early stages of web mashups, there was really little incentive to standardize or share. People would push things out to some degree. But there wasn't any sort of substrate, if you like, for the growth of any sort of broader community around particular domains. The mashup frameworks are starting to change all of this. So the mashup frameworks are there to provide development environments, hosting, and publication. So all of those things that I said were missing when I talked about them on the previous slide. There's a common interface structure. There's the opportunity for building a community, although we'll argue that there are problems with that later on. And the question to ask is this the appropriate spot for scripting for scientists? It can certainly overcome the programming barrier. But, of course, I mentioned before that bench scientists can do great things if you give them the appropriate primitives and the appropriate glue. Well, this is the appropriate glue. But what we need to do now is to give them some of the appropriate primitives. And that's at the level of the individual blocks or components, which is something we'll start to talk about. And the advantage of mashup frameworks is that we can still, if necessary, if we can't find a way of doing something conveniently, we can, in most cases at least, go back to JavaScript under the hood and actually do something sophisticated. So we'll talk about that as we go. So the mashup frameworks are the domain of minor players like Microsoft and Google and Yahoo. So they're not going to go away anytime soon. They're obviously here for the duration. Those of you who've never seen a mashup framework before, I will show you one from Microsoft, which is a thing called PopFly. So a very quick demo. So what we have here is the Microsoft PopFly mashup creator. At the left-hand side, we have a bunch of components sitting in the block bar, and they're organized into categories, and we can drag them. And the reason that this sort of environment is particularly attractive is the ability to come along and click and link up component blocks in that very, very straightforward fashion. What I have at the right is a thing called live image search. If I were to just execute that by itself, it would produce a whole bunch of pictures of Vincent van Gogh, because that's the default search term. On this occasion, I have user input, which is one of the simple tool blocks. And if when I run it, it will just come up with a very simple sort of thing here, inviting me to enter some text. On this occasion, we will decide to go with our host. And on this occasion, when I execute it, it comes up with a whole bunch of things, including Fat Tony from The Simpsons. Now, I'll leave why that happens to a whole bunch of other people, but what you've got is the ability to link these things together in a very, very straightforward sort of way. And if I then come back to edit them, I can come down and I can say, well, in the display here, I can find something which is a moving slideshow, and I can take that and I can get the Ken Burns effect in a matter of seconds as well, just linking that up, running this thing, and of course I get, in a moment at least, the same sort of search requirement. I then execute it. And you get something quite sophisticated coming back fairly straightforwardly and done in a matter of seconds. So if we come back to our presentation, we've got opportunities for mashups from all of the main players. Now, what we've done is to look into all of them, and at this stage at least, PopFly offers a couple of advantages, because whilst it provides that sort of simple interface and glue that you saw in the simple example before, which is something that Pipes does extremely well, it also has the facility to create blocks in JavaScript, and it provides a very good sort of pathway for generating blocks quickly from WSDL descriptions. And in the bio domain, that's particularly important. And so at this stage, PopFly certainly offers some advantages over the other two. But whatever we do with mashups, there are limitations. Okay? The major limitations in mashups are that mashups are designed to do an awful lot of data manipulation, provided we don't actually have to pass large chunks of data between the blocks in a mashup. So they're very, very good at passing an index to the data, and the ideal example of this is, of course, the latitude and longitude in any sort of GIS kind of kind of mashup. Because what I'm doing, of course, is passing what's really just 6 or 7 or 8 bytes of data, and as a result of that index that I'm passing around, I'm manipulating whole gigabytes of data in virtual Earth or Google Maps or whatever it happens to be. So what you see here in each of these situations is the client web browser is doing some sort of limited interaction with the mashup server, but lots of back and forth interaction with the third-party services. So you can have lots of data happening over on this side of the diagram, but relatively little sort of interaction in data passing happening over here when we've got the actual mashup server itself. So BioMashups, we're arguing, sits at the middle ground that I identified before, corresponding to scripting. So it's ideal when there's some user intervention needed. It can also be seen in many respects as a prototype for workflow. We can see ourselves in an exploratory way linking tools together and then deciding, well, yeah, I want this, but I want it in a more robust fashion, and then taking it away and implementing it properly in one of the other systems. It's also the sort of thing where it is a tool which can be a niche for sorting out some of the problems that we have when doing searches to interfaces which are very complex, which frustrate experts and drive students to exasperation. So people who've gone looking for particular biological resources may have experienced that sort of problem. So here are a series of examples. We'll begin with a thing called SDLM, that's the Self-Directed Learning Module 1. This is drawn from one of our undergraduate biology courses. I don't really care about this example as such. It's a very standard sort of thing. There's nothing incredibly interesting about it, except for the fact that it's the standard sort of thing that one would do in an undergraduate bioinformatics class. And the major issue on this side is the series of websites down the right-hand side of the slide, which the students would have to go through to actually execute this thing. And so with this simple self-directed learning module, where we do a blast X on the sequence, look at best hits, and then find out some information about the proteins and so on this occasion, we're able to have an alternative for the students where instead of spending half an hour cutting and pasting from particular sites, they are able to take a series of existing PopFly blocks and link them together to do the same sort of job. We've already trialed this in an undergraduate class, and it was extremely successful, and we're trying to extend that out over the coming year. But more seriously, we'll look at a case study in analysing proteins. So Felicity Newell, who's joined our team, is a PhD biologist in molecular biology with a lot of experience in kidney disease. And so what she did as a study was to look at blocks or creating blocks to integrate services which might be of interest to people with an interest in proteins. So there are two sort of categories here that we go looking for: protein characteristics, looking for the name and the sequence, the journal articles, cross-references and so on, and protein prediction, so with the molecular weight, the isoelectric point, secondary structure and so on. So these are using a wide variety of databases and services, as we can see. So UniProt through NCBI to the DNA Databank of Japan through to KEGG, as we can see on the slide. So the architecture of this slide, of the mashups, is really pretty straightforward, although you may have some trouble seeing that on the screen there with the arrows between them. But you produce 13 custom blocks, a couple of input and output blocks, and then a whole bunch of blocks for processing protein characteristics and for processing protein prediction. So these are actually available on the PopFly site, okay, where she's got a series of example mashups. Okay, this is linked from our site, and of course descriptions of each of the blocks that she's used. So this is just a seed exploration in what can be done using using mashups for protein analysis. So let's look at a couple of examples then. This one is a protein characteristics mashup. Given a protein ID, the UniProt ID, how much can we find out about a particular protein? And we see the structure here where we have a user input protein block, which is sort of a version or a newer version of the standard text input that I showed you before, sending it through the protein information blocks to get various services, PEP-stats, signal IP, and then of course unifying them at the end. And so we can execute this. It takes a minute or so, but it's a fairly straightforward sort of exercise. It loads up in the PopFly interface. We see that there's a default, and we can execute that. Okay, and we'll go back to the slideshow and come back and look at this a little in a moment. Hello. Sorry, we had a glitch there. And on this one, we have the more elaborate sort of problem of, given a protein sequence, what properties can we readily determine from the web-based prediction services? So this requires that you actually insert an extensive protein sequence, and we'll do that in a moment, or we'll have a look at the mashup in a second. Okay, so these are the results from the BioMashups for proteins. This is the protein characteristics mashup. So as you can see from the UniProt protein ID, we get the full protein name, the protein sequence, the features of interest. A little bit further down here, a whole bunch of journal articles and PEP stats and signal IP and so on. And whilst it's not terribly impressive visually, the issue here is the fact that linking those blocks together and executing them in this way is essentially saving about half an hour's work. And of course, with 13 blocks of this kind, people are able to tailor these things to their particular needs. The other one which we look at then is this protein sequence one. It takes a little while longer to run, so I won't actually do that now. But basically, you take a protein sequence and dump it in here. Perhaps, and then execute it, and it comes back with protein characteristics and additional predictions about the protein. Perhaps equally importantly within these frameworks, of course, is the ability to then go over here and to click on the rip this. And rip this is, of course, code for steal. So you're able to take someone else's mashup and to bring it into your own mashup creation area, and then, of course, to immediately take this existing mashup and customize it by deleting links or adding in other blocks from the palette at the left. Now, at this stage already, you can see if you want to find the protein mashup blocks, then of course you can come in here and just do a search on proteins, and you get a whole bunch of proteins, most of which are written by Felicity at this stage. Okay, so that's an example of how the mashups can be used for a particular domain. We can see that the combination is straightforward. We can see that it's something that we can readily do. But it's in thinking about where biomashups might go, it's important to recognize some things for which those sorts of technologies are suitable and some things for which they're not. So whilst predicting is difficult, especially when it's about the future, we can we can see pretty straightforwardly that mashup frameworks can and will support ad hoc exploratory bioinformatics, what I've called here index-based routine computation, so provided you are not passing around too much data between the mashup blocks. And then what I say, they're building, and then the brackets, enclave communities. So this is one of the problems that even something like Popfly will deliver to us, because whilst it will enable us to create a community of biomashups around Popfly, for example, it won't necessarily allow us to build a broader biomashups community. So it's within Popfly. If there are people doing this stuff, there aren't too many at this stage, but if there are people doing this stuff in Yahoo Pipes or Google Mashups, then it's more difficult for them to migrate across. And that's something which we're trying to address through the My Experiment environment by creating a biomashups group to allow people to share these things across the domain boundaries, if you like. There are certainly varying levels of success in allowing scientists and student-driven mashups and the sharing of reuse of components, but that's something which really depends upon the establishment of a community. It will certainly be a long time before mashup frameworks are used for routine processing of high-throughput sequencing machines. That's just not going to happen anytime soon. It's not the sort of thing where we'll see them necessarily processing large-scale collections, although some limited explorations in this area seem possible. And it's very unlikely that in the near future they'll beat Taberner and Kepler at provenance. And whilst predicted as difficult, for us it's really a pretty straightforward exercise. So we have a series of tools which we've developed in Silverlight. Okay, and so these are a bunch of tools such as this Silver map for exploring similarities between genes and proteins. So you're able to see the organization of genes and proteins according to the similarity distance, and to rapidly, at least normally rapidly, reconfigure these things as you go. No, my apologies, I should have double-clicked. And we also have a Silverlight-based genome browser, which rapidly allows us to go between differing scales, okay, and this is all being done in real time from our server. We can go from the full genome level down to local areas very rapidly. And we also have some tools related to a bioinformatics pattern description language. There's a thing called BioPatML. And so these are all written up. So with these sorts of things, you're able to load patterns from the particular pattern repository, and then to bring them in and edit them and then use them as the basis for searches on genomes. So all of these sorts of things, once the relationship between Silverlight 2 and Popfly becomes a little bit more stable, then that's something which we'll find ourselves doing fairly rapidly. But in the longer term, what we really need to do is to build a more general biomashups community. And so we need to see crossover between the mashup frameworks. We need to see some sort of seeding of the community with reusable components in order to get us towards critical mass. So we've started to do that and are encouraging other groups to produce their own blocks. And so the obvious target for that is the My Experiment group. And what we're doing from the other direction is bringing biomashups to the curriculum, where I think that they have rather a large future as part of the new undergraduate biology. Some links here to our group, the Makuta Bio group, our BioMashups pages, the My Experiment BioMashups group, and also to the protein mashups, which I've just looked at before. And this link down the bottom here with the WMV file, that's Felicity talking through some of the mashups that she's created. So acknowledgements to QUT, Microsoft Research, and the Queensland Government, and I'd be happy to take any questions.